everyone. This is our second episode in our research talk about the scientific method. And today I have the pleasure to have our recent PhD graduate, Askhat Myngbay, who will share with us his experience. And welcome, Askhat, to our conversation. And,
thank you very much for inviting me.
A pleasure. Askhat, let me ask you first, who is Askhat Myngbay? What is your background? What made you think about doing a PhD in Azerbaijan University?
Well, first of all, it starts from the school times. I was loving the biology subject and I was interested. I have participated in school Olympiads and competitions and also did some projects related to plant. And my passion grew till, I wanted to be in medical science. Also, I want to be a surgeon initially, the medical surgeon, clinical surgeon. Then by the time I changed the medicine to the biomedical science, starting from the UK, I did my bachelor's degree in UK, then came back to Kazakhstan and did my master's degree. And then I thought maybe I should, further my knowledge and applied for the PhD in Nazarbayev University.
Is there any person that was your role model to do the PhD? Your family, your near group of friends or colleagues or former professor?
My previous, the lead supervisor, Dr. Vyacheslav Adarichev was my role model because I loved his approach to the science, how he did the science and how he, we many times discussed every experiment and did in detail. And I was actually, the interest in the PhD maybe grew with the conversation with him.
I see. And in your thesis that you just defended successfully, by the way, congratulations again.
Thank you.
What was the problem you were trying to address?
My problem was that the diseases like rheumatoid arthritis are the not well-known diseases. We don't know the pathogenesis. We don't know the exact etiology of that disease, and those kind of disease needs further studies, obviously.
And what was the gap that you felt? What was done before? And what was that gap that you felt and your supervisor felt? We need to fill this gap.
Well, there is a mechanism in the pathogenesis of this disease within a joint cavity. And we don't still know how the, I will go to the scientific words, we don't know how the panos is formed within a joint. I mean, we know how it starts. I mean, it starts from the lateral side of the synovial cavity, but we don't know what's the trigger to that formation of panos. Based on the studies on the mice models, we came to know that the CTHRC1, the protein that we looked in our study, is could be the main precursor of the formation of that panos within the joint.
I heard you mention the could be, so it's kind of a hypothesis that you established, right?
Yes, we started with this hypothesis, and I think we established it somehow.
But before we go to the results, which is very important, of course, I want you to tell us what was the methodology you followed to test that hypothesis to prove that this was true or false.
Okay, so every study in the biology starts from the mice models, and based on the results that we obtained from the mice models, We did the immunohistochemistry of the joint tissue and saw that the significantly high concentration of this protein within a joint. Then we wanted to test this hypothesis, our hypothesis, on the human samples. That's why we collected every clinical parameter of the human patients, like we collected peripheral blood, we collected synovial fluid, we collected every clinical parameters like the complete blood count, biochemical parameters, and all those.
I see, Scott, during your explanation, you mentioned the word we. As I know, and we know the PhD is your work, but you have to work with a team where supervisors and peers are important. But also, you had also, as I know, a good stay in another lab outside Kazakhstan. Could you please give us a brief overview of what was that experience you had abroad for the experimental part of your thesis?
Okay. I participate in the, if I understand correctly, you mean the GSK, GlaxoSmithKline in UK?
Yeah,
right?
Well, I meant part of that, yes, because most of our PhD students, as you know, they have international experience. They go abroad for weeks or maybe months sometimes to do part of their work.
I was lucky because first of all, I was granted with that stipend to do my part of my PhD at the site of GlaxoSmithKline, one of the biggest pharmaceutical companies in the world,
right?
And based in the UK, Stevenage. This could not be possible without the NU support. Nazarbayev universities, particularly the school of, at that time, School of Engineering. There was a committee, and they agreed to give the money support or financial support to do this within the 6 months. And
Askhat, now going back to the results after you got your experiments completed, what can you say is the major contribution of your work?
Well, my major contribution is this protein that we found as a marker of the rheumatoid arthritis disease activity can serve and can enhance first of all, the diagnosis of that disease. I mean, diagnosis during the disease, prognosis and controlling the disease. Because as we know that not every treatment strategy is effective. And especially with the rheumatoid arthritis patients, it's very hard. And it's important to control this progression or regression of this disease. And this marker is the marker of the to monitor the disease activity.
And in your case, could you please summarize what is the result in publications from your work?
Okay. I have based, on this topic, I have 2 publications. One was, I wasn't in the, I was within the authors, but I wasn't the first author. That was related to the mice model experiment. And the second publication, where I was the first author, we studied the human samples.
Askhat, I want now to remark the quality of these journals where you publish. What is a typical journal where a PhD student should aim to?
Well, obviously, the Q1 journals, Q2 journals are very good.
Okay, just for those who don't know about it, Q1, Q2 are the quartile in the Scopus or Web of Science index that people try to publish. Are very, very well known indexes. And Askhat, now regarding the future, how do you envision the future work? If you have to continue the work, what would you do next to enhance the work you did?
In my future plan is to do the postdoc, and obviously to continue this work, because this work needs further analysis in terms of the function of this protein within the pathogenesis of this disease.
And now, based on your experience, and what would you recommend to young researchers that are, let's say, doing their master's or even their bachelor, or even doing the high school? What do you think are the typical characteristics of a person that should be embracing a PhD in the future?
If the new generation wants to leave something behind himself, I think he should do the science. He should do the PhD, because the PhD is an opportunity to leave, to leave something behind himself. That's a big opportunity, I think.
Great words to conclude our conversation, Askhat. I want to thank you again for having this conversation and for being part of our PhD program and being
a
good example for other students to follow. Thank you, Askhat.
Thank you. Thank you, Louis. Thank you.